This assignment is for General Biochemistry at the University of New Hampshire, Manchester for Professor Hunter. Today we're going to talk about oxygen transport within the body. Oxygen is important for cellular function because it's the final electron receptor in the electron transport chain during ATP production. The medical term for a failure to meet cellular oxygen demands is hypoperfusion, but is also commonly called shock, and leads to significant tissue damage and even organism death. In humans, diatomic oxygen molecules are physically transported from the atmosphere to the cells by a specialized connective tissue called blood. Human blood is composed of around 50% erythrocytes, or red blood cells, known for their biconcave appearance. Red blood cells contain hemoglobin, a globular protein with a quaternary structure capable of reversibly binding to the oxygen molecules that have diffused into the blood across the alveolar membrane. Within each hemoglobin protein is a coordination complex with a central atom of ferrous iron. This complex is known as heme. The ferrous iron atom in the center acts as a site of reduction and oxidation for diatomic oxygen molecules. The heme group is held in place by the hemoglobin protein through a histidine residue bound to one of the iron coordination sites. As the blood cell travels through the body, environmental conditions around the blood cell create conformational changes within the structure of the hemoglobin protein. These changes in shape cause the amino acids to move around inside the protein. This movement, in turn, affects how oxygen enters and binds to the ferrous iron inside the heme complex. As we discussed before, the heme complex is held in place from beneath by the F8 histidine residue, so called because it's the 8th residue in the helical segment F. The E7 histidine residue is perched above the heme complex and creates a steric interference during conformational changes. In the closed form of hemoglobin, the E7 is pushed closer to the iron atom, thus blocking oxygen from binding. As the protein relaxes into its open configuration, the E7 histidine moves away, allowing diatomic oxygen to oxidize the iron into ferric iron 3. Because E7 histidine is still present and generating steric interference, the oxygen molecule is pushed into a 120-degree angle, causing an imperfect bond with the iron. As the hemoglobin works its way from the lungs to the tissues of the body, it slowly takes on the closed configuration again, pushing the E7 histidine into the heme and breaking the ferric oxide bond, thus delivering the oxygen to the tissues. As the red blood cell travels from the lungs to the tissues of the body, the shifts in environmental conditions cause changes in protein shape. Because the lungs draw in air from the atmosphere, the lung tissue tends to be convectively cooled. This cold is then conducted into the blood, which causes a conformational shift in the hemoglobin towards the open configuration. One of the functions of the human respiratory system is to help balance the blood pH by blowing off carbonic acid in the form of carbon dioxide. This means the pH of the blood in the lungs tends to be much higher than the rest of the body. This change in pH causes further conformational shift towards the open configuration. 2,3-bisphosphoglycerate is a byproduct of glycolysis, with the specific function of regulating hemoglobin oxygen binding. When BPG interacts with hemoglobin, it forces conformational change towards the closed configuration. BPG is generated in significantly lower levels within lung tissue, which allows hemoglobin to achieve a fully open configuration for maximum oxygen binding. As the blood travels out to the tissues of the body, metabolically active organs, such as muscles, will conduct their heat waste into the blood, raising the temperature and causing a conformational change towards the closed configuration. This pushes some of the E7 histidine closer to the heme complex and releases oxygen from the red blood cells into the tissues. As blood travels further away from the lungs, metabolically active tissues will have generated more CO2 via cellular respiration. CO2 combines with water to create carbonic acid, which lowers the pH of the blood. This causes continued conformational change towards the closed configuration, and even more oxygen is released to the tissues. Finally, as tissues release more 2,3-BPG via glycolysis, the hemoglobin achieves a nearly fully closed configuration, and most of the oxygen is delivered to the tissues. Blood then cycles back to the lungs and completes the process again and again. So what effects can alterations in allostasis have on oxygen transport? Well, we describe this using something called the oxyhemoglobin dissociation curve. So along the vertical axis here, we have the relative attachment to hemoglobin, measured as a percentage. And along the bottom axis here, we have the oxygen available to cells, and this is measured by millimeters of mercury and is described as PO2. And in medicine, generally speaking, if a patient has a PO2 of greater than 80, we would consider that to be healthy. And along the vertical axis, we can measure this in the field using a pulse oximeter, which gives us a relative percentage of hemoglobin, oxygen attached to hemoglobin. So one would think that there's a linear relationship between the percentage of oxygen attached to hemoglobin and the oxygen available to the cells. However, because of the cooperative nature of the quaternary structure of hemoglobin, we actually see an S-shaped logistic curve. Now, if we were to measure up from the 80 PO2 to the curve, we see that we have a nearly perfect percentage of oxygen attached to hemoglobin. This is around 95%. This is considered healthy. If we measure up from a PO2 of 60, we see that the percentage of oxygen attached to hemoglobin drops slightly. 91% is concerning, and we would provide supplemental oxygen to these patients. If we move down to a PO2 of 40, we now see the beginnings of a precipitous drop in oxygen saturation, and 75% is very concerning, and these patients would be considered very sick. Now, as we had discussed before, changes in environmental conditions alter how oxygen is attached to the hemoglobin protein. And so, as temperature increases and pH declines, or acidity increases, and the production of BPG increases, this generally occurs closer to the tissues of the body, and we can see that the oxyhemoglobin dissociation curve shifts to the right. And this causes a tendency for oxygen to be ejected as it flows towards the tissues of the body. And likewise, when the temperature is lower and the acidity of the blood is lower and the production of BPG is lower, like when it's closer to the lungs, oxygen has a tendency to stick to the hemoglobin. And this is how we can use the oxyhemoglobin dissociation curve to describe alterations in the affinity of oxygen to hemoglobin. So over the last few minutes, we've discussed how red blood cells use hemoglobin proteins to transport oxygen throughout the body. We discussed hemoglobin and how it uses iron within a heme complex to reversibly bind oxygen molecules. We discussed histidine residues holding the heme complex in place, and also either allowing oxygen molecules to bind or pushing oxygen molecules off, depending on the conformational changes. We discussed how changes in temperature, acidity, and concentration of metabolic byproducts affect the conformational shape of hemoglobin, and how this complex system allows oxygen to bind at the lungs and then be delivered to the tissues. Thank you for your attention.